Firegene Gel Clot TAL Endotoxin Testing Demonstration. In this video, we show how to perform control standard endotoxin (CSE) validation using the Gel Clot TAL reagent assay. Endotoxin testing is essential for ensuring the safety of injectable drugs, biologics, and medical devices. The Gel Clot TAL assay detects endotoxins through a clotting reaction triggered by TAL or LAL reagents. Before starting the experiment, prepare the required equipment. 1. Micropipettes. 2. Gel Clot TAL reagent. 3. A shaker or vortex mixer. 4. A 37-1C water bath. 5. Pyrogen-free reaction tubes. Ensure that all procedures are performed under pyrogen-free conditions. Next, prepare the TAL reagent. Add 1.1 ml of pyrogen-free water into the TAL reagent vial and gently mix until the reagent is completely dissolved. Now prepare the control standard endotoxin. Open the CSE vial carefully and avoid introducing any external contamination. Add 3 ml of pyrogen-free water into the CSE vial. To reconstitute the endotoxin standard, mix gently to obtain a homogeneous solution. After reconstitution, the endotoxin concentration is 5 EU per milliliter. This solution is recorded as the primary CSE stock solution. Dilute the 5 U or mol CSE stock using pyrogen-free water. To prepare a 1 U or mol working solution, this solution will be used as the starting dilution. Perform a series of twofold serial dilutions, starting from the 1 U or mol solution to generate a complete endotoxin concentration gradient. During each dilution step, gently pipette up and down. To ensure the solution is fully mixed, proper mixing is essential for accurate dilution results. Add 100 microliters of TAL reagent into each reaction tube, labeled A through H. These tubes will be used for endotoxin detection reactions. Next, add 100 microliters of each CSE dilution into the corresponding reaction tubes. These tubes represent the experimental group. Prepare 2 additional tubes as negative controls. Add 100 microliters of TAL reagent and 100 microliters of pyrogen-free water. After adding all reagents, vortex each tube gently for 3 to 5 seconds to ensure the reaction mixture is fully combined. Place the tubes vertically into a thermostatic water bath at 37 C. Incubate the reaction for 60 minutes. During incubation, the tubes must remain completely undisturbed. Avoid vibration or movement to ensure proper gel clot formation. After incubation, remove the tubes carefully and invert each tube 180 degrees. A firm gel that remains intact indicates a positive reaction, while a flowing solution indicates a negative result.